Okay, hello everyone. Welcome to this new NeuroSpin Monday seminar. Today we will talk about the BIDS format, which is a standardized format for sharing neuroimaging data, which is great for data sharing and also interoperability between softwares. And today we have three speakers who are researchers and engineers from NeuroSpin: Yann Leprince, Isabelle Dangian, and Sophie Herbst, who will present the BID formats both for MRI and MEG. So we will start with Yann. Yann, that's your turn.
Thank you, Florent, for the introduction. Can you hear me correctly?
Great.
Okay, thank you. All right, so I will try to give you a very brief and general introduction to BIDS without going too much in the details of the spec. So just a few words about myself for those who don't know me. I was recruited last year in April as a clinical data science engineer in the UNIACT, which means my role is to support the clinicians with all the methodological aspects of data processing. And in that frame, I'm very much interested in having a more standard way to organize the data, and this is why my interest in BIDS has spawned, let's say. So the first thing is, what is BIDS? BIDS stands for Brain Imaging Data Structure, and you can learn everything about BIDS by going to this website. At its core, BIDS is a standard for organizing neuroimaging and behavioral data. And by organizing, I mean this ranges from the file formats, specifying which file formats should you use, how should we name the files, how should we structure the directories of a data set in order to facilitate its reuse and its sharing. The benefits of such a standard are numerous, and this goes from being able to reuse your own data set that you acquired a few years ago, and you may not remember exactly how you structured them unless you used a solid established standard such as BIDS. And the benefits also are quite obvious in terms of being able to reuse data that's shared by others for your own work, or vice versa. When you publish some data as open or along with a publication, it makes it much easier for others to reuse the data set. One other advantage of using BIDS is that it will be compatible with many analysis tools, so you will be able to test many approaches on your data without having to write custom scripts to adapt the tools to your data organization. Now, where does BIDS come from? The seminal paper was published in 2016, but in fact, the roots of BIDS are older than this. And BIDS was heavily inspired by the format used internally by the OpenFMRI repository, which is now known as OpenNeuro. So before I go on to describe the basics of the data structure, we need to make a few definitions clear. BIDS is primarily a spec that deals with the raw data. By raw data, I mean data that has been very minimally pre-processed, no more than file format conversion or anonymization. Any pre-processing beyond this, such as motion correction, bias correction, or otherwise, is no more the raw data. It is called derived data. And the derived data has to be kept separate from the raw data. This is a very core principle in BIDS. It has a few practical advantages in that you could make your raw data read-only and then be sure that is not going to be modified incidentally by the tools that you are going to use to process the data. And in order to create the raw data, you have to start somewhere, because typically we do not acquire data directly in BIDS format. We acquire DICOM files on the scanners, E-prime files for the fMRI experiments. This is called the source data. And so BIDS recommends to use this file structure to separate the source data, the raw data, and the derivatives in 3 separate subdirectories of your dataset. The source data is not much constrained by BIDS. It doesn't say anything, well, it does not say much about how to structure it, because by definition, the source data is heterogeneous. The raw data is really the part where you have to stick to the BIDS specification to structure it. And then the derivative data should be structured with one subdirectory for each pipeline that you run on your dataset. And the BIDS specification explicitly says that each of these derivatives may or may not be a BIDS compliant dataset, because BIDS cannot cover all the variety of existing tools. So you are allowed to make a non-BIDS compliant dataset as a derivative of a BIDS dataset, and still call the whole thing a BIDS dataset. All right, so now I will present very briefly the directory structure that is used for a BIDS dataset. At the top level of the dataset is one subdirectory for each subject in the study. And the name of that directory is sub-subject identifier. Inside of each subject directory are multiple subdirectories for the different data types. In BIDS, the data type is a somewhat broader concept compared to the modality. Modality would be T1 weighted, T2 weighted. Both of these modalities belong to the anatomical data type. So here you have, for example, anatomical diffusion weighted imaging, field mapping. There are other data types, and I will list them later on. Inside of these subdirectories are the actual data files. At the top level of the BIDS dataset are a few files that describe the dataset as a whole. The participants.tsv in particular is a table that contains very general properties of the participants, such as age, sex, handedness, clinical scores, clinical status, performance status, etc. The dataset description.json is required by BIDS, and this must contain very general metadata at the level of the whole dataset. The name of the dataset, maybe a version, the list of authors, the links to the paper that describe the dataset, the DOI of the dataset itself, if it has been published, the license, any information that is useful for those who want to reuse your dataset. After that are the usual good practice files, README for user-readable introduction to your dataset, and changes, which is a change log of the changes you introduce in each version of the dataset. Now that I've described the directory structure, I want to go into a bit more detail and describe how the file names themselves are formed. So here is an example of a file name from a BIDS dataset. It is made by three parts, and I will start by the last one, the extension, which is a very standard thing describing the file formats of the file. Before the extension is a suffix that usually designates the modality. So it could be T1 weighted, T2 weighted, bold, diffusion weighted image, or many others, which are MRI modalities, but it could also be something derived that is derived from the raw data, such as a defacing mask, which is very important to store when you perform defacing to anonymize your data. And the first part of the file name is a chain of key value so-called entities that are separated by underscores. Each entity contains a key separated from the value by a hyphen, also known as a minus sign. And these entities are really the key attributes that differentiate this one file from every other file in the dataset. So taken together, the entities and the suffix can designate a piece of data uniquely across the whole dataset. This is actually a general rule that is enforced by BIDS. Every file must be identified uniquely by its name alone, even when you take it out of the directory where it belongs. So if you take the file out and send it to a colleague, you can still know where it has to be stored in the BIDS dataset. So for example, you are not allowed to omit the subject entity. The subject part will really be part of any file name across the whole dataset, at least any file name that pertains to a particular subject. There's a few other rules, such as the ordering of the entities is fixed by the standard, so that a file that has a specific set of entities has a file name that is defined uniquely. And for each type of file, the specification will describe which entities are required, which are optional, and sometimes which are disallowed. Okay, I want to now take a small detour for the case of multi-session data, because we often acquire data multiple times from the same subject in the frame of a study. For example, the longitudinal study, we have to represent this in the data set. The way this is done in BIDS is by adding one level of nesting, and as subdirectories of the subject directory, you will have session directories. So in this example, you have two sessions, one that is called pre-drug and one that is called post-drug. This also means that you have to add the session entity to all file names that belong to a particular session. You can also use this file, this per subject file, underscore sessions.tsv, to describe the key variables that change between the sessions. If you do longitudinal imaging with different ages, you have to put in this file the data that you would otherwise put in the participants.tsv. And because adding the session layer changes the whole hierarchy of the data, some people prefer to always include the session level in the data set, even when they acquire only one session, in provision of maybe later on acquiring other sessions and adding them to the same data set. Okay, so this is all I would describe about the directory structure. All the detail is in the spec, which is actually quite readable. I will now move on to the 5 formats. BIDS requires you to use certain 5 formats for certain types of data. Actually, 3 of them. So first, it will require you to use Nifty for the images, with compressed Nifty being recommended. It requires you to use TSV, tabulation separated value, for tabular data. This format is really similar to the more common CSV format, except tabulation are used instead of the comma, and every spreadsheet program is able to read and write these files without problem when configured correctly. So these files are used for the participants.tsv. They are also used for storing the events of a functional study, the stimuli, the responses. They are also used to store physiological recordings when that is acquired. And the last format that is required by the spec is a bit special. It is the JSON format, which is used by really at the core of BIDS to store metadata. JSON, for those who don't know it, is a really simple file format that is able to store key value dictionaries. And in BIDS, it is used in particular to store the metadata that does not fit in the regular data files. The Nifty files, for example, have a header which is able to store some basic data like voxel size, maybe a transformation to the scanner-based referential, but usually not much more than this. Using a sidecar JSON file, that is a file with same name but a JSON extension, you are able to store much more metadata, and in particular all of the metadata that usually would require you to go back to the DICOM file to read the metadata. You can store it in these sidecar JSON files. And this is actually done automatically. Isabelle will describe you the tools that you could use to convert your data. They will write these JSON files for you. Okay, so now at present, BIDS specifies the following data types: for functional MRI, diffusion-weighted imaging, field mapping, anatomical imaging, MEG, EEG, intracranial EEG, and pure behavioral data without associated imaging. For each of these data types, the standard will list which files are required, which are optional, but you may come across the case where you have a piece of data to store that is not specified by the standard. And in fact, BIDS is an evolving standard because our methods keep evolving all the time. So the standard is evolving in the spirit of an open source project. You are able to make proposals, and there are different communities around BIDS who are regularly proposing enhancements to BIDS in the form of BEPs, BIDS enhancement proposals. You can find the list of these BEPs at this page, and there is, for example, one for quantitative MRI. There is one for positron emission tomography, which is currently in the final phase. It's considered ready by its authors, and it's being submitted to the community to give feedback before it can be eventually merged into the BIDS spec. So when new things are integrated in BIDS, the version of the standard is increased. New minor releases will contain backwards compatible changes, and at some point the community may decide to make breaking changes, and that would belong in a future version 2.0. Okay, last two slides. I want to quickly mention a few tools that you can use to work with BIDS dataset. The BIDS validator is a very precious tool when you are preparing a BIDS dataset. Its job is not to do any kind of data processing. It's just to verify that your dataset is properly formatted according to BIDS. There is an online version which works in your browser. I should say it does not send any data to the server. It works only locally, so your dataset will stay private. And we also have a command line version of the validator that is pre-installed on the workstations of NeuroSpin by the IT group. And last, I want to mention a few tools that allow you to work with BIDS formatted data. There are some tools that were designed from the scratch up to work with BIDS. These two are actually from the Poltrack group, MRI QC, to do quality control of T1 and functional imaging. fMRI Prep to do the full pre-processing pipeline of functional images. And I mean, in all the tools of the community are more and more becoming compatible with BIDS. There is one effort in particular I wanted to mention. This is called the BIDS apps. And these consist of containerized processing pipelines in the form of Docker containers that you could run with either Docker or Singularity. They contain the typical usual tools from the community bundled in a container and adapted to work directly on BIDS formatted dataset. So I will stop here and hand the floor over to Isabelle, who will present you how you can convert your datasets to BIDS in NeuroSpin.
Hi everyone, do you hear me? Can you
Yes.
show my screen? It's okay for my screen?
Yes. And we see your screen.
I'm sorry, F5 don't start. Okay. So thanks Jan for this very clear introduction to the BIDS standards. If you don't know me, I am Isabelle Danguin-Courcol and I work in the UNICOG laboratory. My job is to help a student, a researcher to use a lot of tools and to develop new tools. And also I am in charge of a lot of web services such as Wiki, the UNICOD website and so on. So today I'm going to speak to you about a script, NeuroSpin to BIDS, which is available at NeuroSpin. So you know now the motivation is to have a common organization and infrastructure for sharing data between people and labs at NeuroSpin, but also anywhere. The script was initially proposed by a PhD student in the Christoph's team. And after some changes, we use it for many years now. Remember, in the BIDS standard, we have 3 levels of directory, and this script works at the raw directory level. In this directory, we use data unprocessed, but data can be converted, such as DICOM to NFT. So just to remember you have sources directory, in which we have very files without any modifications, such as EPROM logs, DICOM, and we have also the derivative level, which all data which are processes and analysis. So this script is mainly dedicated, designed for MRI data. We will speak with just after with Sophie about MEG, and we have had some discussion also with for AG modality. So this script is one possibility, is not the only one, of course. So what is important at the end is that your dataset is conform with the standard, and for that you can use the BIDS validator. Here on this slide, we have a summary about steps performed by this script. It's very basic script in which we can interface a lot of tools. So the first step, the idea is to create a list of data you want to retrieve from scanners at NeuroSpin, and you want to give to create specific names for the BIDS dataset. So for that, we will use a specific file, which is participants to import.tsv. I will give you some details just after. And on this file, we have a lot of information to know what you want to import and what information in order to create the specific file name for BIDS dataset. So very technical. Here you have the path, an example of the path where you can find data for MRI. It's NeuroSpin acquisition database, and you want a lot of a list of names such as BIDS dataset, sub1, ANAT, and so on. So the second step is to create specific files for the standard. Do you remember we have mandatory files such as data description.json, in which we have a small, just a few information such as the number of the version of BIDS, and for example, the name of the BIDS dataset. But you can have a lot of information if you want. And the other one is the participant.tsv, in which we have the list of participants, and for each participant we can have a lot of information. And we have also the possibility to add optional files, like changes, readme, license, and participants.json. It can be very interesting to add this one because we have the description of fields are mentioned in this one, in this file. And for that, we can use a very basic function in Python, and we can use also a Python module which is specific for BIDS, and this module is mini-BIDS. So the next step is to import and to convert data from DICOM to Nifty format. For that, you have an interface with a command line, which is available at on the network. This command line is DCM to Nifty batch. It's just to convert DICOM to Nifty and to create also JSON file for each DICOM. For this, when you use this command, you have to create a specific file. It's automatic. It's created by the script. It's a batch DCM to Nifty.yml, in which you have the list of input and outputs what you want to use. Then you have an optional step. It's the possibility to deface your T1. You know, when you want to export your dataset outside of NeuroSpin, you have to deface, to remove all information about patients or volunteers. And for that, we select here for this script the pdeface module. It's a Python module in which we have FSL registration with a specific template in order to remove specific parts such as face and ears. So it's an automatic registration. So it's very interesting to check, to have a manual check at the end to see if any parts of your brain that you want to process are not removed. And at the end, it's optional, but you can do just after the importation, but you can do what you want. If you add a new information in your data set, you have the possibility to run this command line, BIDS validator, in order to check and to have a summary about your BIDS dataset. So you have a summary, for example, as you know now, sorry, you have a summary with the number of subjects, which are modalities are available in your dataset. And also we have the possibility to see warnings and errors. For example, warnings can be something is missing if you have fMRI files, and if you don't have events, you can have a specific warning for that. And an error can be a problem in the file name. For example, if you have fMRI files and you don't have the bold modality in the file name, you can have an error. So now I want to explain in detail the preparation of the importation. So it's a specific file to the script. It's participants to import.tsv. And as you see, it's a TSV file, tabular separated value, in which you can have all information in order to retrieve and to import, convert all data. Here you have an example. This file is open with OpenOffice, for example, but you can use any editor file. You can also use a command line such as a VI, for example. So the idea is to be sure to, when you use this kind of interface, to have the correct value here to indicate how your values are separated. In this file, we can see the first row with the name of the column, and we have information specific to the infrastructure at NeuroSpin, such as NIP, or for example the dates or the location. Here we have the name of the scanner, but it's also possible to have a path. And on this file, we have also specific information for BIDS. Here we have the subject label. We have also here the possibility to add a session label. We have also the possibility to add acquisition label. And here we have a set of parameters in order to build the file name at the end. Here, for example, we have func, it's the modality, and after we have a long part of the file name needed for BIDS. We have task, key value. To remember, we have key and value. So task and the value is log standard. We have the possibility to add a run, and what is mandatory is a bold to indicate the modality. Here you have specific examples for NeuroSpin. For the first importation here, we have the path available at NeuroSpin in order to retrieve data. And here we can see the number of the seconds. And to indicate with the script which seconds do you want to retrieve, you have just to indicate this number. And you can see the number is this parameter. So here, the second line is to have an idea about the pattern for the BIDS specification. So some parts are mandatory and other ones are optional, such as the session. So here we have to construct something like the sub and participant label. So we have the name of the here we have the row level. After we have the subject level. Here you have CES for the session, the modality ANAD, and the data what you want to import. We have also the possibility to add run for fMRI. Here we have, no, for the last one, sorry. Here you have an example with the run. So in at NeuroSpin, we have a path, something like that. We have a specific number for the sequence. And when you want to add a run for the fMRI modality, you have optional key value here. And for example, for this example, we have this part which is already mentioned in the TSV file. Now we have a summary of steps for this script. First, you have to create a specific directory in which we want to import your data. Then you have to create exp info in which we can create the TSV file to indicate what you want to retrieve. So you have to fill this you have to fill in this file, sorry, and just you can do that with command line, for example, by using VI or another editor tool. So after that, you have to launch importation by using Python and the script. If you want, some options are available, for example, to import the events. And when the script is launched, you can answer to iterative question in order to create a lot of files which are needed for the BIDS dataset. So those files are data descriptions, image, engines, and so on. And you can select if you want to deface T1 data, you have the possibility to say yes or no. And at the end, you have the possibility to validate your BIDS dataset to be sure that all is conform with the specification. Here, when the importation is done, we have 3 directories. The first one corresponds to your BIDS dataset. You can see here the structure, the organization of a BIDS dataset. It's the row level. And we have also the exp info directory in which we have the file used to indicate what you want to retrieve, but also the batch. Do you remember the batch underscore descent2nifty.yml, in which we have the list of name files, and this file is used by descent2nifty batch. And we have also a third directory, which is report, in which we have the download. And in this file, we can visualize if data, the list of data are imported. So, for example, if data is not found or already imported, here we have the information at this level. And here we have in this file report BIDS validation.txt. We have a copy of the result of the BIDS validator. As you know, as you see, the NeuroSpin 2 BIDS implement just a part of the whole BIDS specification, mainly for MRI, because at NeuroSpin, I think it's very pretty easy to import data coming from the scanners, because the infrastructure and the organization is very robust. So now probably when you want to import and to complete your BIDS dataset, probably you will have specific needs. You can have to import phenotype information, physiological data, or I don't know, perhaps a lot of information. So you have the possibility to complete with your own script the dataset. If it's a little bit difficult for you to read and to understand the specification, we can work together in order to create a very specific script in order to have a complete BIDS for your experiment. Here we have a list of links. Very interesting to read. So the first one is the BIDS specification. It's not so difficult to read the specification. I think it's very interesting to take a look to understand deeply how it works. Here we have Open Neuro. It's very interesting to take a look also because you have a lot of data set on it, and you can browse a lot of data set and you can understand by visually by to see an example is very interesting to understand how you can have a whole data set. And we have also neurospinwiki.org in which we can have explanation to use this script, and also you can go to the NeuroSpin Unicog to retrieve the script. And the last slide, it's about the web services, which is available at NeuroSpin and proposed by Antoine Gregis. This service, you have the possibility to use dataset which are available at NeuroSpin, but you have also the possibility to add a new dataset, a new project, if you want to share data here at NeuroSpin. So that's all. I leave the floor to Sophie.
Okay. Can you see my slides?
Yes.
Okay, great. So I'm Sophie Herbst. I'm a researcher in the Cognition and Brain Dynamics team at Unicog. And in the team, we have an ongoing collaboration with the MNE Python developers. And today I'll talk a bit about the MEG standard for BIDS and tools you can use, and some efforts we've been following in this collaboration to make the tools more usable, and especially with NeuroSpin datasets. So if you listen to what Jan was saying, there's not much of a difference for the MEG BIDS compared to the fMRI or MRI BIDS. So MEG BIDS was, the paper was published in 2018, but it was actually initiated in 2016 at BioMag, and there are now BIDS standards for MEG data, EG, IEG, and they are highly similar because the data are very similar. And it's supported by some of the main processing toolboxes like Brainstorm, Fieldtrip, MNE, SPM. It can handle the standard MEG file formats, and there's also a validator tool to check if your conversion worked. And in the bottom right here, I put the link to a talk by Guiomar Niso, who is the first author of the MEG BIDS paper and highly involved in the ongoing discussions about this at Live MEG last year. So if you want to hear an inspiring talk about this, check it out. Slides are not moving. Okay, so this is a picture from the paper where you see the layout of a typical MEG study folder. Can you see? I hope you can see my mouse. So you have on the top left, you would make a directory for your study, and just as for the MRI, you would have subject folder with the session for each recording, basically. And in there, so then you have the modality specific folders. So you can have, first of all, you would have the MEG data or the MEG folder, but you can also store the MRI. If you did an anatomical scan, which we often do for source reconstruction, you can have behavioral data and also a sessions file. So in the MEG folder, what would you have? So what I marked here in red is the MEG data file as it comes out of the machine. So at NeuroSpin, we have an Electa machine, so our files are in FIF format, and that's really the raw data without anything else being done to it after the recording. And then with that, you can have coordinate files where you have the position of the fiducials and the HPI, so the head position indicator calls that help you locate the head of the participant while they're in the MEG machine. You can also have some photos, for example, where you put those markers when you prepare the subject. You can have digitized head points that you did when you prepared the subject. You can have a list of your scans. Then you have the so-called the MEG JSON file, which is a sidecar file that has all the information about your recording. I'll go into a bit more detail on the next slide. Oops, not yet. You have a file with the channel, so that's a TSV file, just like a simple table. And you can have the events, that is the triggers, so when you send your stimuli. I'll show that also in a bit. Then in the anatomy folder, you would have typically the T1 that you recorded. And in the behavioral, well, that's the standard for behavioral data, so you can have the events and some descriptions thereof. And then other files that you would have in your study folder is a folder called empty room, where you can store all the recordings. So generally when we start on a new day or a new subject comes in, we record a minute or so of the machine without the subject to get the noise of the sensors, and you could store these in the empty room folder. You would have a list of your participants, a general description of your data set, and then importantly, the derivatives folder where all the pre-processed data goes into. So to give you a bit more of an idea of what's specific in the MEG BIDS standard, so you have this MEG JSON file where you have, for example, the name of the task. So you could put, I don't know, if you have different tasks, you could put that, or you say empty room or resting state. So basically whatever you ask the subject to do during the recording. And then you also have a lot of recording parameters that are important to know. So the sampling frequency, the position of the DVAR, if any software specific filters were applied during recording. And then some basic information, for example, if you have the landmarks or how many channels you have. Yeah, all of which are important if you want to know what has been done with this recording. And then another important file is the events table. So this exists usually closest to the data. So you would have it for each participant, each session, and each run. And here you store the, basically what we call the triggers. So your markers that you send during the recording and are also in the data. So you have the timing with respect to the start of the recording. Here you have the duration of the respective stimulus. You can already give some names, which is of course very useful. So if you have condition names, you can directly give them here. This is the original trigger value, and this is the sample with respect to the recording. So mandatory are only the onset and the duration, but yeah, I think it's very helpful to put in this additional information. So people who want to look at your data set actually know what has been done. And here's an example of a data set. I'll speak about this a bit more later, that has been recorded at NeuroSpin and that is now available at OpenNeuro in BIDS format. So you can go, you can check it out and see that you have all these files, and yeah, you can see how they look. So how do I get there? There's a lot of information to put into these BIDS structures, right? And luckily you're not on your own, but all of the standard data processing toolboxes have nice tools available. And here I speak about the MEG Python tools because that's the ones that we use. So there is a Python package called MEG BIDS, and it comes with a lot of functions that already do all that stuff. So you don't have to make all these files manually. So you just have to feed in the information that you have. And for example, you can use the write raw BIDS to write the BIDS data set, or the write ANAT to put the MRI in. And just to give you a bit more of an overview, so that would be the write raw BIDS function. You basically give it your data, you set up your BIDS path, you can give your events, you would give the names of the events and so on, and then it takes care of setting up this structure for you nicely. And importantly, there's also functions to update, especially the sidecar function, sidecar files. So for example, this data set description file or MEG JSON, you can update them later on if more information becomes available, or if you realize you forgot something, which is important because you don't want to destroy your nice structure by making any updates that are not compatible. So the typical BIDS workflow for MEG would be you record your data, you get your machine-specific output format, and ideally you set up your BIDS repository right away. You validate that the first import is correct, and then you would build your pipeline directly on that. And all the pre-processed files would go into the derivatives. And yeah, as I said, you would update the sidecar files if necessary. And then the anonymization, it's a bit of a tricky question at what point in the pipeline you want to do that. But you could do it, for example, when you want to publish the data. So you dephase the MRIs. You see an example of a dephased MRI. If you have already done your co-registration, and importantly, then you would have to remove all dates and participant info and so on. But there is also functions for that. So more specifically at NeuroSpin, I think the when to put your data in BIDS is really an important question. And ideally, we do that as early as possible to avoid having multiple copies of the same study. So we are already working together with Isabel to come up with a conversion script that can import the data from the acquisition server, just as she showed for the MRI. And then you can also build your pipeline directly on BIDS. So you wouldn't want to build your pipeline on a repository that you made and then just before publishing convert everything to BIDS, because you might have some incompatibilities with the file names. Then, yeah, I already spoke about the anonymization before. I wanted to give you a little more insights into this collaboration I was talking about. So we have a grant together with Alex Cranfort at INRIA, Max Chaumont at the ICM, Virginie, and Richard, who is the engineer employed on this grant at INRIA, where we're actively working together to make the M&E Python software more BIDS compatible, and share data sets and pipelines from those institutes, and try to make them more versatile, and especially with respect to BIDS. And I want to show you that in a bit more detail. So we're working on the M&E study template, which has come a long way. So it has originally been published in 2018. Then it has been translated to BIDS by Stefan Appelhof. We have done several hackathons, as you see in the upper right, at the Cognition and Brain Dynamics team. And now Richard has done a lot of work. So this is a pipeline that is not part of the BIDS standard, but it runs on BIDS formatted data sets. So it's very useful. Once you have your data in BIDS, you can do all kinds of all the pre-processing steps basically, or decoding, time frequency transform, and even source reconstruction. So I wanted to show you that. I hope you can see that. Now I can see my tabs. Great. So yeah, it lives on GitHub. So I put the link, and as you see here, you have some folders. So for example, if you look into the pre-processing folder, you have all the steps that you need to do: import max filter, frequency filter, make epochs, run ICA, and so forth. And Richard has built a very elaborate website. I'm sorry, my Zoom is blocking my. I can't see my. Oops. Here's the website. So if you go here, you'll have a lot of explanations about the study template. And the most important part of it is that there's a config file, which is called config.py, where you have to put all the specific parameters that you want to use. So just to give you some ideas, it runs on MEG and EG, so you could put the channel types. You can put the references for EG. You can put the montage of the cap. You can put channels to be excluded. Did I end up on the MEG template, EG template here? So basically, the idea is that you just have to update this config file. You don't have to touch the real scripts. And if your data is formatted already in BIDS, it will already read a lot of this information from the BIDS data. And to show you an example, so this data set that I showed you before, we have run it through the study template. So here it is. Here you have the configuration file specifically for this data. So you see it's much shorter. It just contains the parameters that you wanted to change. For example, here you have the filters, or you do some resampling. And then another important point is that it generates these elaborate, that's probably not the most elaborate one, elaborate reports of everything that has been done to the data, like ICA cleaning, for example. Here you see the ICA components. And here you would see the report of a subject that has been fully processed with this. So you have the covariance matrices. You can check what has been done to your data, outlier removal. You see the basic evoked responses, the topos, and so on. And all of this is already included. So you just have to set your parameters. And that's the data set that is shared. That you can, if you want to play with that and you don't have your own data yet, you can download this data set, for example, and run it through the study template, or use some of the other examples. So I think that's all I wanted to say.